concept is that our brain has a certain range of size, which in women of this age group, the range of normal is pretty large, about 500 grams, and the mean is about 1230. Males in the same age groups, quite a big range, and the mean maybe 140 grams more. So when we say a brain is atrophic, just weighing it doesn't really necessarily prove it, because you can see that in a male, you could have an 1100 gram brain, or in a female 1000, and that's just a statistical range. The way we like to diagnose atrophy, this is the entire brain, looking at it from the side. When we see gaps between the gyri of the brain, you see these black areas that are basically space. Gyri are separated from each other, and then when you look at the brain sliced sagittally, you can see that these gaps persist. In a normal brain, those gaps between gyri, which are called sulci, are very small, even on the surfaces. So the most reliable criteria for me is the morphology of the gyri and sulci. Now, people with advanced Alzheimer's disease, for example, can have brains in the 850 to 900 gram range. And so sometimes the weight confirms the visual impression. So what do we mean when we say a neurodegenerative disease? Well, in the gray matter, what we mean is that the neurons are progressively dying, and then there will be secondary changes in the white matter tracts that are connected to the neurons. In most neurodegenerative diseases, it's selective. It's not the entire brain. So the areas of the brain most severely affected give us a clue as to the probable etiology which this means cause. What's the causative thing? But there's a common theme that resonates among all the neurodegenerative disorders. And it goes like this. If a neuron makes an abnormal protein, or too much of a normal protein, the cell becomes stressed. And sometimes the cell is able to degrade that protein, use up the amino acids, and stay alive. But there comes a point at which abnormal proteins aggregate so much in the cytoplasm of a neuron that our ubiquitin proteasome system cannot degrade all of them. And they lead to death of the cell, usually by apoptosis, but it's basically the cell's inability to degrade these abnormal proteins. Now, why is that important? Well, first of all, that sort of gets at the molecular basis of these diseases. But histologically, these abnormal proteins look like inclusions or visible aggregates in the cytoplasm of neurons, and that's kind of how we make the diagnosis of what's going on, because it reflects the underlying biochemistry. So here's the concept. How does the cell process an altered, or in other words, misfolded protein? This is the protein that comes out of the Golgi complex with aberrant structure, all right? Maybe the amino acids are in the right sequence, but it's not folded properly. So normally, when we have normally folded proteins, molecules called chaperones make those proteins, here are the peptide chains being produced in the endoplasmic reticulum, and these chaperones pick up these and cause a certain folding, and that's a good thing. That would be a protein that we can use for normal functions, properly folded. Oh, by the way, you're going to get a complete copy of this handout, so if you just want to take notes on my voice. Now, when that protein becomes old or denatured, which is generally what happens to all proteins in cells, we have this beautiful system that we call the ubiquitin proteasome system. And these ubiquitins attach to the dying or old protein via various ligases. It shoves that ubiquitin protein complex into a structure called the proteasome, and the proteasome degrades that entire structure into peptide fragments, and then the cell is not harmed, and the peptides are reused, and everything's happy. Now, here's what can happen if, when making these, trying to make folded proteins, if an abnormal protein comes out of the rough endoplasmic reticulum, and this can be due to viral infections, genetic mutations, other sorts of things. I'm getting feedback. Are you hearing feedback? Okay, I'm going to assume it's all right. We can create an excess amount of misfolded protein. It doesn't have to be a genetic mutation. It can be, but there can be other processes going on that makes too much. Now, here's the cell's remedy for this. The cell will automatically, this is called the unfolded protein response, it'll make less of this protein, turn down translation of the protein so there will be less to deal with, and it will increase the synthesis of these chaperones, which can then guide it into the proteasome system. So make less and get rid of more. If that is successful, the cell will live. But if there's too much of the bad protein, the cell undergoes apoptosis, programmed cell death, and that's how neurons die in the neurodegenerative disorders. They end up undergoing apoptosis. So, here are some of the diseases and the proteins that accumulate. In Huntington disease, it's a Huntington protein. However, if your gene that encodes the Huntington protein has an expansion of these three nucleotides, they're called tag repeats or trinucleotide repeats. They're encoding a region near the end terminus of the protein. All of us have a certain number of repeats in our gene that's encoding the Huntington protein. It will be different for different people in the room. But if we don't exceed a certain amount of those repeats, we will never make too much abnormal Huntington protein. So it's considered a mutation. It's called a mutation when we expand the number of these repeated triplets of trinucleotides in the gene encoding Huntington protein, and when they get above a certain number, the protein that comes out is abnormal. And it accumulates in the cells, in neurons, but weirdly enough, this particular Huntington protein accumulates in the caudate nucleus and the putamen, not everywhere. Neurons of those two areas. And this causes the disease called Huntington disease. Have anybody ever seen somebody with Huntington disease? Okay, so Woody Guthrie, the famous folk singer, had Huntington disease. It tears your heart out when you see somebody with Huntington disease because they have these abnormal involuntary movements that are violent, and then the next thing that happens is they get dementia as they get older. So it's a horrible disease. Now, Alzheimer's disease, same general process, accumulation of a misfolded protein, not due to a genetic mutation, except in a small number of cases, which we call early onset Alzheimer's disease. In the vast majority of human beings that develop Alzheimer's disease, it happens beyond the age of 60. The incidence increases as you go to the 70s and 80s and 90s, and it's not a mutation. It's just too much of an amyloid precursor protein. We'll show you how that happens. Parkinson, the abnormal protein is alpha-synuclein, and that accumulates in the substantia nigra of the midbrain.
and sometimes it accumulates in other portions, other regions of the brain. So why these proteins have a proclivity for certain anatomic sites is interesting, but I can't explain it. So that's some basic science. Now we're going to put on our clinical hat for a little bit. What is dementia? Dementia is not a disease. Dementia is a symptom and sign of an underlying disease. So there can be 5 different diseases all resulting in a patient who has dementia. So here's the definition produced in Harrison's Internal Medicine, and this definition really hasn't changed. It's an acquired deterioration of your cognitive abilities, and how bad? They impair the successful performance of activities of daily living. You can see this is a subjective call. For example, I won't talk about you, Dr. Kraspolsko, I'll talk about myself. I lose things at home and can't remember where I put them far more often than I did when I was 30. All right. I forget things that people told me a week ago, even though when I was 30, it would have stayed fresh in my RAM. I could, my memory, my short-term memory is not as good as it was, but I still know how to dress myself. I still know how to drive my car from home to Wright State. I can still give a pathology lecture. I can think. It's just certain parts of my cognition are not as swift as they used to be. That's not dementia. Dementia is when it impairs the performance of my daily activities. So for me, if I couldn't teach because I can't make sense when I talk to people, I need to get out of here, right? That would be dementia. So it develops over months to years, and here's an important thing, at least 2 different domains of cognitive function. So just memory deficits alone are not enough, and typically this is the one that tells us executive function deficit means you can't carry out a task that you used to be able to carry out, such as I get in my car, I drive 15 miles, and I end up in the parking lot here. If I can't find my way, I have a problem with executive function. Okay, so just losing memory is not dementia. We all don't process information as rapidly when we get older, and that's just called the deficit in short-term memory to various degrees. My mother is 97 and she remembers everything bad that I ever did. Memory like a steel trap. Okay, so here's the different etiologies of dementia. Alzheimer's disease, neuropathologic changes, far most common. Here's probably the second most common. This is a disease of your blood vessels in which the small arterioles are narrowed, or let's say partly stenosis is another word, narrowed. They don't let as much blood through. That's arteriolosclerosis. Degeneration of just the frontal and temporal lobes. Different than Alzheimer's, different process, but that's probably number 3, and Lewy body may be also tied for number 3. So when I do autopsies for people with dementia, 95+% of the time it falls into one of these 3 categories, and Alzheimer's is the dominant one. Here are some other diseases that can cause dementia, including HIV, prion disease, Huntington disease, uncommon causes. So this, I like history of medicine. So in 1906, Alois Alzheimer presented a case to the German psychiatrist. The case consisted of a person who was delusion and jealous, paranoia, and memory loss, and the process started at age 51 and ended at age 55. And that's his original concept. He called that, after himself, a cause of pre-senile dementia. Well, I would submit that delusions and paranoia are not typical of dementia. Those are probably into other psychiatric disorders. But the point of it is that this happened in an older person. And then a couple of people several years later, 70 years later, said that this is the cause of senile dementia in the elderly, which happened a lot. And so even though that wasn't what Alzheimer described, his name became attached to this concept of dementia. And that became a highly prevalent disease and remains so today. So this is the most common cause who are over the age of 65. We want to make that qualification. Early on, you just see some impairment of higher functions. Usually, episodic memory means remembering what happened yesterday at 5 o'clock. What was I doing? As opposed to all my childhood memories are intact. Later, you lose your executive functions, become disoriented, you become difficult to interpret space and what you see. My mother has this now. When she watches a television program, she will turn to me and say, "What are they doing?" She can't interpret the visual images, the language, judgment, etc. Eventually, if Alzheimer's disease takes its terrible toll, people stop talking. They can't even recognize their spouse or their children. They don't care about taking a shower. They become sort of curled up in a place, and then eventually they refuse to eat. And so the typical person that I see at autopsy, their body looks like this. They're wasted. Usually, I'll give you an example. The last female I did who had Alzheimer's disease weighed 81 pounds. She was 5'4". So this is kind of the incidental finding, but I just wanted to show you that the NIH did a study looking at how does the BMI, body mass index, relate to death from any cause in over 500,000 people. This was published in 2006. And I find this interesting. They restricted their 500,000 people to people in this age group. So I would call this older, but not extremely elderly. And they said, what is the relative risk of death among these people? And, you know, they followed them for several years. And what they found was that a medium, kind of in between BMI, is the least risk of death. We all know that if you have a lot of excess weight, it takes years off your life, but it's also the very skinny that takes years off the life. And unfortunately, the Alzheimer's disease patients go from in here somewhere back to here. So just on that basis alone, they're at increased risk for dying. They're basically starving themselves. It's rare before it's 50, but if we do have somebody with Alzheimer's disease less than age 50, we call that early onset Alzheimer's disease. And the chances are that person has a germline mutation that they inherited from mom or dad that led to this early onset. The prevalence doubles every 5 years. So I'm just a little older than this group, so I made it through the portion of life when I would have had early onset Alzheimer's disease, and now I'm headed toward a progressively increased likelihood that I will develop this disease if I live, by God's grace, to be in the 80 to 80, 85 and 9, 40, I will have a 40% chance of having dementia due to Alzheimer's disease. Those are scary numbers. And so we have an epidemic of this disease in our country because our people are living longer. Less than 10% are familial. Now here's something really important. Most physicians will put the label of Alzheimer's disease on a patient who has dementia, and the imaging studies, especially a high-resolution magnetic resonance scan, show atrophy predominantly in the temporal and frontal lobes. And they will say, this is characteristic of Alzheimer's disease. And they're right, it's characteristic. But it could just as easily be frontotemporal lobar dementia or degeneration. It's just that the odds are it's Alzheimer's disease. So they're making a probability statement when they make it a presumptive. The definitive diagnosis is by pathologic examination of the brain. Now, our testing pre-mortem is getting better and better, and tests done on the blood. Dr. Kraspolska gave me this sort of a release thing that there's now maybe a blood test. And also, if you take cerebrospinal fluid out of people, you can test their cerebrospinal fluid. And those can give you, let's say, clues to the presence of Alzheimer's disease. But currently, most informed neurologists, geriatricians, pathologists would say you need to examine the brain, which is why people still send me patients for this purpose. Now here's a nice progression of how good this MR scan. So here's a normal brain by MR. Notice the ventricles are small. The gyri are pretty full and robust. And these little black areas are air. That is tiny cracks between these nice, robust, thick gyri. Things are covering up. There we go. So grade 1, the ventricles are a little bigger. We call that hydrocephalus. And the gyri are a little more narrow and the sulci are more wide. Grade 2 gets worse. Grade 3, really bad. And grade 4. So you may say, well, you know, I think grade 4 looks like grade 3. Yeah, these are interpretations by expert radiologists, but the point is that the degree of atrophy gets worse as you go through the process of dementia. The two things that they go by the most are atrophy in either the frontal or temporal lobe and the amount of hydrocephalus. Now, why is there hydrocephalus? Anybody have any idea? Why are those ventricles, which carry cerebrospinal fluid and circulate, why are they getting bigger? The reason is they're just filling up the space that the brain has undergone atrophy. There's no increased pressure in there. It's just they're expanding because the brain is shrinking. So here again is a brain. This is a normal brain. And this one shows cortical atrophy. Notice how wide these sulci are in the temporal lobe and in the frontal lobe. As you get back to the occipital lobe, that's usually spared in Alzheimer's disease. And here it is on cut section, normal. Look at these big ventricles. Look at the gaps down here in the temporal lobe. And here, so this is the region of the hippocampus. And down here we're seeing called gaping horns. We call it gaping temporal horns, meaning increased space between the gyri of that inferior temporal lobe. You're talking here or everywhere? Yes. Oh, yes, yes. I see what you're saying. Right, there's a nice demarcation. And in Alzheimer's disease, if you in fact would do the measurement of the thickness of the gray matter, it would be decreased. This photo is a little overexposed, so that's contributing to a little bit, but you're absolutely right. The gray matter is shrinking. All right, so the structures of the medial temporal lobe, which is right down here, specifically medial inferior temporal lobe. Here's the hippocampal region of that medial temporal lobe. Here's the amygdala. Those are the places that are hit hardest and earliest in the early stages of Alzheimer's disease. And later on, the frontal and then sometimes the parietal lobes. Major microscopic abnormalities are neuritic plaques, and I think the best way to describe those is that they are a tangle of dendrites and neurites. So if we say that normally we have the afferents coming into the cell body of the neuron and the efferents going out, those structures, instead of being nicely organized, become all tangled up and abnormal. And it has to do with what's going on in the neuron itself. Now, this particular plaque it's a graveyard of a neuron that's gone. So this big hunk of stuff in the middle is not the nucleus of a neuron, it's amyloid that has aggregated. That was the unfolded protein that caused the death of the neuron. And so what's left is that unfolded protein, and there'd be no neuron right at this spot. Here's a neuron, no neuron here. Now, there are neurons that live, they're not dead yet, but they have accumulation of abnormal proteins in their cytoplasm, and they develop these long tails. I'll show you why it's called a tangle later. But these are neurons that are probably going to die, but they're dying by a different mechanism than the ones forming the neuritic plaques. Here's a silver stain. Every one of these little spots, these little brown spots, are extracellular aggregates of tangled neurites, afferent and efferent, and they're taking up the silver. This was the classic way that we identified these neuritic plaques by silver stain. Here it is on higher power. These are normal neurons, these large black things in here. These are our neuritic plaques, and some of them have a central core of amyloid. Yes, sir. You know, that's a really good question, and I don't know the answer to it, because I don't try to classify those when I'm examining a brain. So that's a great question. Somebody probably knows, but I don't. So this particular dead neuron didn't accumulate too much amyloid, so you don't see a big blob of black, but this is usually a central amyloid core, and the way we prove that is we do a special stain, Congo red. Amyloid takes up this red dye intensively, and you notice how red that is. These are the glial processes of the astrocytes. They take it up very minimally. And then if we really want to get specific, we can do an immunofluorescence in which we take an antibody against the amyloid protein, incubate the tissue section with the antibody. The antibody has a fluorescent tag on it. You look at it under the microscope, and then you can see the green coloration, which means amyloid was present where the tagged antibody attached. Here's another silver stain showing a neuritic plaque. They can be variable size. They're usually surrounded or admixed with microglial cells, which are the resident macrophages of the brain. And then they have some reactive astrocytes at their periphery. Here's how we detect it now. The silver stains were nice, but they're technically difficult to do. I used to have one histolab that would try to do the silver stain, and they weren't doing it for other specimens, and so they'd try to do it on my brain sections. Sometimes it would come out way too black, other times way too pale. But this immunostain, which is now what the University of Pittsburgh does for me, the Brown reaction product means that amyloid beta protein is present wherever there's brown. I won't go into the details of the stain. But here's the funny thing. By silver stain, I used to look at a section, I'd say, there's about 20 plaques here. When you do the immunostain, that number is like 40. In other words, you can't see them all. You certainly can't see them on a hematoxin and eosin stain. Silver makes you see more, and this really detects any deposit. Like this little one right here, you'd never see that on an H&E or silver stain. They're most abundant, as we said, in the medial inferior temporal lobe, and they usually spare the motor and sensory cortices. So people with Alzheimer's disease do not develop motor or sensory deficits early on. Now, they may be weak because they're not moving and they don't have a lot of, let's call it volition, where they want to do things, but they don't have a defect in their motor or sensory cortex. Here's the dominant protein. It's called amyloid beta. Here it is on the silver stain. Here it is on H&E. The only time you can really see it on H&E is when it clumps up into a big clump. And then it looks reddish purple on H&E, but honestly, I don't even look at the H&E sections to diagnose this because they're just too insensitive. So here's our component, amyloid beta, and this is a peptide that is derived from processing a normal protein in the neuron called amyloid precursor protein. So this is not a mutation, it's just an alteration of a normal protein. There are two species that we call amyloid beta 40 and amyloid beta 42, and this, as you might expect, just two more amino acids in the amyloid beta 42. Some really nice experiments done in the 90s have shown that amyloid beta 42 is more toxic than 40. So I gotta tell you a story. I was going through these ideas during a exercise in which we call team-based learning with the med students. This was 10 to 15 years ago, and one of the students raised their hand at the time and said, Dr. Colas, you're describing these two amyloid beta peptides, but I have learned that amyloid beta 42 is more toxic than amyloid beta 40. I didn't know that. I said, where'd you find that out? He said, I did the research with other researchers in Boston, and he had published that in the New England Journal of Medicine before he came to med school. And I said, well, shut my mouth. Like, thank you for sharing that, because, you know, this required a lot of work to determine that a protein with just two more amino acids is more dangerous. I mean, that's really good experimental work. Sometimes you can get definition or deposits of these peptides, and the surrounding neurites won't be destroyed and tangled and messed up, but that's the exception. So I'm going to stop here and go slowly and ask if there are any questions before we move on, because we're going to get real biochemical here. Yes, sir.
Oh, you said it wasn't a mutations for those beta proteins?
No, these are not mutations. They're caused by abnormal processing of a normal protein.
Okay.
Yeah. Yes. You got it. And I like the point you made, which I forgot to make, and that is that all of us have some of these abnormal peptides in our neurons. Alzheimer's disease is a matter of quantity. How much of this abnormal protein do you have in your neurons, and how many neurons have died as a result of accumulation of these proteins? If you do sections of brain of people my age, you're going to find amyloid beta in their brain. It just won't be enough to correlate with the presence of dementia. Thank you for making that point. So, any other questions before I do this? Because this slide tells you sort of the basis of all the pharmaceutical research that's been going on to try to treat Alzheimer's disease. So we know that pharmaceutical research is driven by a very deep understanding of molecular biology, what's actually happening. So here we go. In a normal neuron, we have this protein here called amyloid precursor protein. This whole thing, it's a transmembrane protein, and that protein can be cleaved at 3 different sites. The cleavage site by one enzyme, beta, alpha, and gamma. If we cleave the amyloid precursor protein at the alpha site, followed by the gamma site, this one is released extracellularly, it's soluble. This one is small, this one stays intracellularly, and this does not damage the neuron. This is happening in all your neurons right now, and it's not a disease. Okay, now watch this. If this very same protein is first cleaved by beta, okay, instead of by alpha, we end up with a piece of amyloid precursor protein that is too long. And it's called amyloid beta peptide. The gamma still works, and the gamma keeps this tail of this cytoplasmic tail of the transmembrane protein in the neuron. But now we've got this thing, all right? And this thing has a tendency, because it's an abnormal structured protein, here's the monomer, then these things aggregate into oligomers in the cytoplasm of the neuron. They start sticking together, and then they become big aggregates, and then they're broken down into fibrils of amyloid. Remember, this is amyloid beta protein, 40 or 42, and then those things form these plaques. And here is another pathway, which I would like to spend more time on, but in the sake of time, I'll just say that tau, which is another normal protein, becomes abnormally phosphorylated, and we get aggregates of tau. So really in Alzheimer's disease, we have dysfunctional amyloid beta peptide and abnormal tau protein accumulating, and these abnormal tau can cause abnormalities with the microtubules. They can aggregate and damage neurons, and these things accumulate in the cell so much that the ubiquitin proteasome system cannot get rid of this much protein, and the cell undergoes apoptosis due to accumulation of these abnormal proteins. So most of the research in pharmaceutical industry was trying to find a way to manipulate this process of cleavage. So trying to find a way to suppress or inhibit beta secretase so that we wouldn't be forming these molecules, trying to enhance the activity of alpha secretase so that we wouldn't form the abnormal amyloid beta protein. And I'll skip this by just saying, I mean, this is really interesting stuff if you like details, but the point of this slide is that what happens in these neurons before they die? The answer is the synapse doesn't work properly. And so you can get neurologic dysfunction in people with Alzheimer's disease way before the neuron dies. And this is kind of the, if you will, the pathophysiology, the abnormal neurophysiology is that synapses are not working properly. So as it accumulates, if we do alpha followed by gamma, I remember that is always good. That doesn't cause the abnormal protein. And it's soluble and it's catabolized and everybody's happy. If we do beta followed by gamma, I call that bad guys. And that creates these abnormal amyloid beta 40 and 42. And it's the accumulation of those abnormal proteins over time that causes the death of neurons. And here's the clinical correlation. Dementia gradually appears when the number of dead neurons becomes quantitatively significant and is affecting certain portions of the cerebral cortex, such as the frontal lobe and the temporal lobe. And this was to your point. Virtually all persons have lost neurons by this mechanism by the time they're over 65, but you won't notice it clinically if the quantitative number of neurons so damaged is smaller. And here's the effects of this abnormal protein. It likes to form aggregates as opposed to the soluble, shorter fragment. It forms these beta-pleated sheets. This is actually the structural definition of amyloid. It's a pleated sheet, and don't ask me what that means. The physicist can tell you, but it's a certain conformation of an aggregated protein. And when it gets in this conformation, it resists degradation by the ubiquitin proteasome system, and it is recognized by the surrounding supporting cells. Microglia are the macrophages, astrocytes are the supporting cells of the central nervous system, and those cells are, if you will, signaled that something is wrong because of the amyloid beta is accumulating. And it's the combination of this response from normal cells plus the neuronal problem that results in toxicity to neurons. And now I'm going to switch gears and say, how do we actually know that this happens? Like, is this all just interesting stuff from animal studies? The answer is no. We have evidence from the genetics of people who have early onset Alzheimer's disease. I'm going to take a break after I show you a few more pictures. So here's what we mean by the neurofibrillary tangles. We've been talking about these neuritic plaques. This is the other finding, and actually a tangle is something that happens in the cytoplasm of the neuron, and it's a bundle of filaments. There are normal neurofilaments in neurons that displace or encircle the nucleus. The nucleus in this cell would be here. In this cell, here's the nucleus. Here's the nucleus down here. These bundles of filaments are aggregating in such a fashion as to sort of push the nucleus aside. And so that's what we call a neurofibrillary tangle. So two things about these. These are not specific for Alzheimer's disease. You can see these in other neurodegenerative diseases, and these are far less numerous than the neuritic plaques. So even though they're important in the pathogenesis of neurodegeneration, we don't count them because we cannot correlate the amount of dementia with the number of neurofibrillary tangles. So they are not as countable for diagnosis as the neuritic plaques. On the other hand, if we see these increase in number, that's additional evidence that process is going on to contribute to the dementia. So where do we find the tangles? Cortical neurons, entorhinal cortex, which is in the temporal lobe. It gives you afferents to the hippocampus, amygdala, which is temporal lobe, basal forebrain, and the raphe nuclei. So notice we keep getting this theme, temporal lobe. Here's the tangles. If you want to see them by electron microscopy, what are they biochemically? They're hyperphosphorylated tau. Tau is a microtubule-associated protein that puts the microtubules together properly. So if we hyperphosphorylate this normal microtubular protein, it messes up the way that we assemble those microtubules, which then make the neurofilaments, and they become that same old theme, aggregates of abnormal protein in a neuron. And this is the biochemists. This was put out in 2007, and this is really down to the actual biochemical details of how the tau gets hyperphosphorylated, and it's kind of beyond my ability to explain, frankly. And here's our neurofibrillary tangles. If we do tau, remember how we did with the senile plaques or the neuritic plaques? We made an antibody against amyloid beta peptide, and when that antibody found amyloid beta peptide around a neuron, it caused an antigen-antibody reaction. Then we just attach a chromogen or a color molecule that becomes You can make it brown or red, whatever you want, at the site where the antibody found amyloid beta. You can do the same thing with a monoclonal antibody against tau. And though, therefore, when we see brown reaction product, we say, aha, that anti-tau found excessive amounts of tau in this neuron. Therefore, this is an abnormal neuron or a neurofibrillary tangle. And as you might expect, that's a far more sensitive way to identify neurofibrillary tangles than just eyes and a silver stain. 3 more minutes. Let's get through this one. Cerebral amyloid angiopathy is almost invariably part of Alzheimer's disease. And this just means that the blood vessels are undergoing deposits of amyloid. It's not just the neurons that are affected. However, this disease can occur without the global neuronal degeneration. But interestingly, it's the same old abnormal protein, amyloid beta 40, and you can even see this in people without dementia or with dementia. So this is a disease that is morphologically definite. You know, I can diagnose it under the microscope, but often it doesn't mean as much clinically. It doesn't necessarily make a person demented. In fact, I would say it doesn't make a person demented, because if your neurons are functional and this is all you have, you're not going to be demented. So we're going to start with familial. Let's take a break, come back in about 7 minutes. That would be 2:05. And if you have any questions, you come on down and talk with me. I'm going to